Okay, you guys, let's go over the HIV example and then we'll go over the cell communication. So it explained a little bit about how HIV uses a little bit of cell communication to actually infect a cell. And so it talked a little bit about the two types of receptors. And so there was the Cd4 receptors. Let me erase this from last hour here. So you have the Cd4 receptor. This is the membrane of a human cell. Oops, it's still a little wet. It's bleeding because it's wet. All right. So here's the membrane. And so there's the Cd4 receptor on the human cell. And then there's also this receptor called CCR5. And so there's these two different kinds of receptors. And it talks about in the picture that when the HIV virus infects the cell, how it does that is it binds to both of the receptors on the human cell. So this is the human cell, which means then that this membrane around the HIV virus has something to bind to this and bind to this. And therefore, what happens is once they bind to both of them, as the picture shows, it initiates pretty much something similar to endocytosis. That's what this picture is showing you here. So it fuses with the membrane, and what it's doing is it's allowing its genetic material to go into the human cell. We'll learn a little bit more later on in the course about how the HIV virus works, but the HIV virus, the genetic material for it is RNA, and that's how it infects the cell, is inserts its RNA into the cell and basically stays in that cell for the remainder of the cell's life. And then that RNA can trigger your cells to make new HIV viruses that can go and infect other cells. So that's how that works. So in order for the virus to actually put its genetic material into the human cell has to have both of these receptors. So it shows you what happens when it's missing, the human cell is missing that CCR5 receptor. If it only has that CD4 receptor, notice that the HIV virus in this picture here still binds to the CD4 receptor, but without that CCR5 receptor, it cannot inject its genetic material into the cell, and so therefore the cell never becomes infected. And the little caption underneath there says that as in resistant individuals, so that there are some individuals that are resistant to HIV, the HIV virus. It doesn't mean the HIV virus can't get into their body. It can get into their body, but the HIV virus, their cells their bodies, the people who are resistant don't have this CCR5 receptor, and so therefore the virus can't infect their cells, and so therefore their cells don't become infected from that. And so we call those individuals resistant individuals. And so, so that's how that works. And so the first question in there, making connections, asks you what would you predict about CCR5 that would allow HIV to bind to it? So what would you predict about it, Devin? Okay, yeah, so shape is complementary and therefore can fit together with the HIV, and so therefore they kind of like enzymes and substrates, all right? They're very specific, so are the receptors with what it binds to. So absolutely. And so so the HIV virus, that's the case for the CCR5 virus receptor as well as the CD4 receptor. So then the second question asks you, how could a drug molecule interfere with this binding? So they talked about drugs and discovering this, they call this the co-receptor, that the HIV needs both of these receptors in the human cell. And so once they discovered that, they began looking at drugs and researching drugs that would stop the HIV from infecting the cell. And so how could a drug interfere with this binding to the CCR?
Like I said, that it would mimic like an HIV virus, and like it binds the site, and then like it wouldn't allow the HIV virus to bind to the cell.
Okay, it kind of like maybe like a competitive inhibitor, kind of like a little bit. So he's saying if the drug bound to that first, that CCR5 receptor, then the HIV virus couldn't bind to it, and therefore couldn't put its genetic material into the human cell, and therefore the human cell wouldn't get infected. Absolutely. Anybody else have a different idea of how a drug could interfere with this happening between the HIV and the human cell? Allie?
I said maybe it could like change the shape so that they can bind together.
Okay, yeah, absolutely. If it changed the shape, if the drug changed the shape of the receptor, then the receptor wouldn't have the right shape for the HIV to bind to, and therefore stop it from happening. So absolutely, a shape change. Joe?
Just wondering, like, does pH have an effect on the receptor?
Yeah, but it would affect, pH would not only affect the receptor, but also lots of other things in the cell itself. So changing pH probably wouldn't, ultimately, it would damage some other things too, all right, in addition to that. Maddie?
Are there any other biological effects for people that don't have CCR5? Or is it just kind of like not really a big thing?
You know, it doesn't seem to be, but I think that this is still kind of new in terms of discovery type of stuff. Because that's another, because they're not targeting the Cd4 receptor. Why are they not targeting that? That's why? Yeah, dangerous side effects, because they do know that this is involved in other pathways, and then if we stop that from working, then we have some additional side effects from that. But to their knowledge, that there's not any like real detrimental side effects for this. I'm not sure if there might be probably some side effects. There is usually with most artificial things that you take in. That's why when you see commercials for drugs on TVs at the end, they say, and they say it really fast, all the things that could go wrong. All right, so there are some side effects, but usually the side effects either when it becomes approved for the drug, the side effects either affect so little numbers of people that it can be a big deal for some people, but for the majority of people it's not, or the side effects are the people would rather live with those side effects versus what they're going on, you know what I mean? So I don't know what those side effects are for this particular, like they gave it a particular drug that, in 2007, but I don't know what those side effects are, but I'm assuming that there are some. Shane, what were we doing? My original question was anything else that...
Well, I was gonna say, like, even they developed a drug that pretty much kills the CCR5, or not kills it, but like makes it detach from the cell or something like that. I don't know the right terminology.
Okay, so actually get rid of it?
Yeah.
Okay, I guess if you could get rid of it, yes, that would do it. So yeah, if they get rid of it.
Do something that would turn off the gene that codes for CCR5 so that it's not produced in the first place.
Okay, so actually turn off the gene. Usually, so everything is coded for in your nucleus, your DNA. Your DNA, sections of it are genes, and the genes produce a protein. And the CCR5 would be an example of a protein produced by your genes. And so if you turn this gene off so it can never make that protein, then absolutely, then you wouldn't have the CCR5 to actually begin with, and then that solves the problem as well. Okay, absolutely. Anybody else? You can see good ideas. All right. All right, so so I just wanted to have you guys read that because it gives you another application of why cell signaling and knowing about cell signaling is important and can have important ramifications in people. Okay, so let's go to the go back to the packet I just passed back to you entitled Cellular Communication. And so I want to go over the answers to that with you. All right, so this one, cellular communication, looks like this. I just passed it back, not the one I just checked off today. And so the first question is actually on the back of the first page. The first page has all the pictures. I'll be going back and forth between those. So number one, I'll kind of go start in the upper left-hand corner and go around clockwise. So number one, the little small circle with the little dots in is your ligand. To the right of that is your cell. Below that, the U-shaped molecule is the receptor. And to the left of that is the membrane channel. So that's what the things are in the pictures. So then number two asks you to describe how the shape of the ligand and the shape of the receptor are related. So how are those shapes related? What do you think, Ryan?
It says the receptor is specific for a particular ligand.
Yep, absolutely. And so they have like a complementary shape that fit together like puzzle pieces or a lock and a key. Number three asks you which of the methods are illustrated on the front page would be between cells that are in close proximity to one another. And there are two answers here. So which one of these pictures would be in close proximity? Nathan? B and C, absolutely. So B is showing them the molecule in the membrane is actually fitting together with a molecule in the membrane of the next cell, so they're actually touching. And then C, that one cell is sending out a signal molecule to the other cell, but because the cells are adjacent, the signal doesn't have to travel very far. So B and C are the answers there. So then number four, which of the cellular communication methods appear to be for signaling between cells in different parts of an organism, or long distance communication? That would be what, Sydney? D, absolutely. And something that kind of alludes to that is the blood vessels. So blood vessels can travel throughout your whole body, so things can move very far distances from one part of your body to another via your blood vessels. And so that's what that's showing you there. Then number five asks you, which of these four communication methods would a ligand need to have the longest quote-unquote life, or be stable or stay in this right shape for the longest period of time? And that should be the same one that has to travel the furthest distance, which is D. All right, so therefore it'd have to stay stable longer that way, meaning in the right shape. So then number six, it gave you four terms and it kind of told you what the prefixes mean, and then had you identify what the different pictures would be in this. So going back to the first page, A, that should be the autocrine signaling system. B is juxtacrine. C is paracrine. And D is endocrine. All right. And then 7 asks you, let me shut this, the communication occurs for lots of different reasons: mating, defense, homeostasis, and it asks you, do all ligands for cell communication have the same chemical structure or shape? And give reasons or examples about why that would be, or to support your predictions. So, we want to take that one? Number 7. Roman? Number 7.
I said no, because each ligand has a specific shape to bind with a specific receptor, and all ligands can't have the same structure, since the cell won't be able to tell one ligand from another, which would alter their cellular communication.
That's right. That's right. So it's like it helps the communication be more specific, so you have specific responses. Absolutely. All right, so then number 8 gives you some different examples, and you had to categorize which one of those types, juxtacrine, autocrine, paracrine, and endocrine, would that be describing. So A, we had talked about with quorum sensing with bacteria, when we talked about that TED Talk and so on, and that once the population gets dense enough, then they secrete signal molecules and so on. So that should be, what do you think? Max, what do you have there?
That is paracrine.
Paracrine, absolutely. So the signal doesn't have to move very far. They're not touching the cells aren't touching, they're kind of close together, and so then the signal is released and can travel quickly between those. B, you have plants that have channels that connect them called plasmodesmata, and so the molecules can move directly from one cell to another through this. And what would this be an example of? What do you think, Abby? Juxtacrine, absolutely. So they're touching each other. And then C, morphogens are molecules that are produced in embryos. And so that helps for the cells in their development to develop into particular tissues or tissue types into different cells. This would be an example of what, Raymond? Paracrine. And then, because they're close together, so the signal doesn't have to go very far. And then D, cells in the human pancreas release insulin when blood sugar levels are elevated. The insulin signals cells in the liver to begin absorbing glucose and converting it to glycogen for storage. So the signal travels from the pancreas to the liver and causes a change to happen there. That would be an example of, what do you think, Joe?
Endocrine?
Endocrine, absolutely. Number 9 also has some examples there. So we have pheromones released by a female gamete cell, which is the egg, that provides a pathway for the male gamete cells, the sperm, to travel, increasing the possibility of fertilization. So the egg cell and the sperm cell, they're far apart, and this egg cell releases this pheromone, which is a chemical signal. And as it releases it, it's in higher concentration right around the egg cell, but then as they diffuse, it kind of diffuses and it gets lower and lower concentration. The sperm cell kind of follows this signal pathway to the egg cell, and therefore knows where the egg cell is to fertilize it. So this would be an example of, what do you think? Josh?
I said juxtacrine because the two cells are relatively close to one another.
Okay, yeah, when they get close to, they do eventually get close together, but they don't start out close together. So this is actually an example of endocrine, because, and I want to point out, because a lot of people think that because of this picture in the front, that the endocrine showed the bloodstream, that it has to be in the blood. Endocrine is just long distance. And so when the sperm cell versus where the egg cell is, it's considered a long distance for that. And it doesn't necessarily have to go in the bloodstream. B asked you about cancer cells release their own growth hormone rather than relying on growth hormones from the host organism or from other cells. And this presents a challenge to cancer researchers looking for ways to slow the growth of cancer cells. So cancer cells, a growth hormone causes growth, which for a cell, that means it's going to divide. We grow by becoming more cells big. So a cancer cell releases a growth hormone, which triggers it to grow and divide. So this, because it's releasing its own growth hormone and communicating with itself, this is an example of what?
Autocrine.
All right, yeah, autocrine. All right, so it's communicating with itself and triggering its own self to divide. And so that's why you can have just one cancer cell, and that cancer cell produces its own growth hormone. So it bypasses, usually we make growth hormones that trigger our cells to divide, but the cancer cell can trigger its own self to divide. So it can divide when it's not supposed to divide, and that divides uncontrollably, and that's what we call a tumor, is a mass of cells like that. All right, then C, neurotransmitters are ligands that are released from the axon of one nerve cell to the dendrite of another nerve cell. And this helps to propagate the signal across several cells. So you have the axon, and you have a little space, and then you have the dendrite of the next neuron, and it's showing you one cell releasing signal molecules, and the other cell has receptors. So this would be an example of what? Savannah? Yep, and you will be right. Absolutely. They're close to one another. The signal doesn't have to travel very far. And then D, cells in the pituitary gland produce TSH, which is a hormone, and then that TSH is received by cells in the thyroid, which is another a gland. The arrival of TSH in the thyroid triggers production of several hormones, which then travel throughout the body to regulate metabolism. So this would be an example of what, Matt? 9D.
Oh, I said endocrine because the TSH cells are traveling through the body.
Yes. All right, absolutely. All right, then 10 and 11 are applications of this, and asking you to think about how, if we want to, 10 is asking you if you want to prevent communication, and 11 is if you want to enhance communication. So if you are a medical researcher and you want to prevent two cells from communicating with one another, so you want to stop that from happening, what could you do and research to do if you want to stop them from communicating? Roman?
I said that a researcher can make a substance that jams the receptor of the cell completely, which will prevent the signal from happening at all. Or you can just make a substance that prevents it from the cell particular signal.
Okay, yeah, absolutely. So we have two things there. So on the adjacent cells, you can get some kind of molecule. So here are your two cells. Here's the receptor right here of the cell. So when this guy here releases the molecule, if you have some kind of molecule that can block that receptor, then this ligand can never bind to the receptor, and therefore these two can't communicate with one another. And then the second thing that he said is that if you stop the ligand from being released in the first place, so either stop it from being produced and therefore released, then they can't communicate with one another. So those are two different ways that you could stop communication or prevent it from happening. Then 11 is asking you the opposite. What if you want to enhance communication? So you want these cells to communicate with one another. How can you promote communication between the cells? What could you do? Katie?
I said that you could make artificial ligands that would do the same thing, or like mimic a normal ligand. They'd release the same signal, so you'd basically just be increasing the concentration of the ligands.
Yeah, absolutely. Absolutely. Shane?
It's kind of like that, but I said make a medicine that's kind of like a catalyst that speeds up the reactions of making ligands so that it makes more.
Okay, all right. So yeah, actually increase the production of the ligands naturally in the cell. Anybody else? All right, very good. And then the last three are some extension questions here. So 11 talks about how cellular communication, we looked at various organisms, bacteria, plants, vertebrates, and then it tells you that the mechanisms of cellular communication also have some similarities among these various species. And to come up with or explain how scientists can use what they know about cellular communication systems to show evolutionary relatedness between species. So how could we use what we know about cellular communication systems in various species to show relatedness? Shane?
How they communicate.
What do you mean?
So like how certain cells communicate with certain cells. also like our sex cells. So like that way they can see like, okay, they evolutionized to be like this in reproduction. We're the same way because they communicate the same.
Okay, so looking at similar processes like
Yeah.
between different species and looking at how communication, you're saying, between like the egg and the sperm cell, how it happens in one species and how it happens in another species, and comparing them, and if they're the same, what does that mean?
It means that like one evolutionized from another or something like that.
It just shows the relatedness.
Yes.
Yes. All right. Yes. All right. Absolutely. Maria?
Kind of similarly, just because a lot of our cells use the same mechanisms for communication, I think that points to the idea that we're all derived from like unicellular organisms that evolved over time and then at one point separated into different species.
Okay. Yeah. Absolutely. Okay. Anybody else? All right, very good answers. All right, number 12. Some hormones like estrogen and testosterone are lipids and therefore nonpolar. Explain why a receptor protein would not be needed for this type of ligand to activate a response in the cell. And more specifically, it should say, explain why a receptor protein would not be needed in the cell membrane for this type of ligand to activate a response in the cell. And why is that, Nathan? What was that? Snow. All right. Nathan was getting ready to answer the question before the snow.
Does nonpolar molecules can pass directly through the phospholipid bilayers of the cell?
That's right. So anytime that a ligand is nonpolar or hydrophobic can go right through the lipid bilayer. It doesn't need a receptor protein, and so oftentimes the receptor proteins for those kinds of ligands are in the cell. They're intracellular receptors. Very good. And then lastly, 13. What are some stimuli that could cause a cell to release a ligand and therefore begin communication with other cells? So what are some things that could trigger a cell to start making signal molecules and releasing them? Sage?
DNA code could be turned on and then be transcribed and translated into a ligand.
Yes, absolutely. Maria?
The presence of a bad bacteria that would require a response so that the cells attack bacteria.
Okay, yeah, the presence of a bad bacteria that the cell would respond to and start releasing something to attack it.
Maybe like chemical changes of pH in the environment.
Yeah, changes in the environment, pH, temperature could cause that to happen. Absolutely. Katie?
A cell is dying and the ligand is released to signal that it needs to be broken down.
That's right. If a cell is dying or something is wrong with the cell, it releases a ligand to, there's actually a death signal that can be released. We're going to talk about that. And actually there's a whole process of kind of cell suicide, kind of like where it kills itself off. Absolutely. All right. And as horrible as that sounds, it's actually a good thing. All right. Because usually it happens when something's wrong with the cell.
I don't know if anyone saw this, but like,
Hey, wait. Shh.
uh,
Joe, Roman, Nathan. Go ahead.
like, if some sort of ligand were like supplemented into the bloodstream and then were to be used as communication between
Is that door locked?
Okay. All right. Yeah, absolutely. It might be locked.
Hello, how are you? I don't want to invite myself in.
Oh, come on in.
I just need Madison Crawford for 30 seconds.
Were you done?
Maybe, I don't know. I was saying like if a ligand were like supplemented or like directly put into the bloodstream, like a medicine you take.
Yeah, and that would trigger.
Yeah, that would be like a ligand that would communicate to cells.
Yeah, absolutely. Sometimes it can be in your home, talking about ligands being produced, it could be a hormone. So a hormone from one part of your body is released, goes to and binds to a cell in another part of your body, and that could trigger the cell to release another hormone or something like that, all right? So that could also work, and like same idea with the medicine.
Do cells work, so let's say you do have artificial ligands, you know, like by medicine. Would a cell recognize like, oh, there's ligands, I'm gonna make some, like the same? Would it make more of the same? I don't know how to put it.
Oh, you're saying if you give like an artificial ligand, and what that would do
And the
is,
cell like recognizes, oh, okay, I should make some too.
not usually, unless that's what the ligand is for. If the ligand, when it binds to the receptor on a cell, it's going to trigger some kind of cellular response. It's going to trigger a transduction pathway in a cellular response. So if that cellular response of that ligand is to make more ligands, then that would happen. So whatever that ligand does, whatever cells it binds to, and whatever response it triggers, that's the response you're going to get as a result of that. Does that make sense?
Yeah.
Okay. All right. Anybody else want to share about stimuli? You guys have great ideas. All right. Alrighty, so then let's get out our notes and we're going to continue on with that.
Cutting off circulation.
Okay, so we are on, I think, 11.4, right? Yep. So let's just get back, let's kind of review, it's been a weekend. So remember cell signaling, there are 3 stages. We talked about reception, where that's where oftentimes if it's in the receptors in the membrane, the signal molecule, the ligand will bind to the receptor, and that triggers the next thing, which is transduction, and that's what we talked about on Friday. So what happens, they call this the signal transduction pathway. So it's usually activating certain enzymes in this pathway, and then eventually we get to our response, which is the change that's going to occur in the cell as a result ultimately of that ligand binding to that. So we are on the response part here. Just a quick little thing about the transduction. We talked about how the transduction can occur with one molecule binding, and then we have a signal transduction pathway and you get a response. But then we also talked about, as part of this pathway, that you can initiate what's called a second messenger. I just want to spend a minute and just review that real quick. We talked about this example here with the second messenger, cyclic AMP. So we have a molecule, a signal molecule binding to the G protein linked receptor. That's your first messenger. And so that's going to activate a G protein, which is going to activate this enzyme called adenylyl cyclase. That enzyme catalyzes a reaction, the conversion of ATP to cyclic AMP. This is called a second messenger. It's what's going to activate your first protein kinase, which then will activate your second, third, and so on and so forth, until eventually it activates a molecule like an enzyme in the cell that triggers your cellular response. So sometimes transduction involves second messengers like cyclic AMP. The second, we talked about 2 examples of second messengers. The second one was calcium being the second messenger. And so this right here is the same idea. The first messenger is a signal molecule that once it binds, activates your G protein, which activates an enzyme. In this case, this enzyme creates a second messenger called IP3, which binds to the gated ion channel in the ER, causing it to open and calcium ions to diffuse out, and the calcium ions act as a second messenger. They activate certain proteins and activate that phosphorylation cascade until eventually you get your cellular response. So I just wanted to point out your second messengers, cyclic AMP and calcium. Did we talk, I can't remember, in some hours I did, this is where we left off, but did I talk about these pumps? Okay, I did in this hour, okay. Alrighty, so, alright, so now we're on the response here. So, there are two places where the response can occur, in the cytoplasm or in the nucleus. So ultimately, a signal transduction pathway leads to regulation of one or more cellular activities. The response may occur in the cytoplasm or may involve action in the nucleus. Those are your two spots, the cytoplasm or the nucleus. Many pathways regulate the activity of enzymes. And so meaning that oftentimes the response that you get is because either an enzyme that's already in the cell is activated, or the response could be turning on a gene that will make an enzyme. Either way, an enzyme is going to be in the cell and be able to be used in the cell, and it wasn't able to be used unless the signal molecule bound to it. So let's look at this pathway here. This looks like a lot of things going on here. Actually, the most of this, all the way up to this, is the same as the picture we just looked at. So let's look at this. It says reception, binding of epinephrine. Epinephrine is a hormone, and it says to a G protein linked receptor, and it says one molecule. So one molecule, one receptor, one epinephrine binding to that. Then that G protein linked receptor activates, so you have an inactive G protein, it activates the G protein. So the G protein activates, which then activates the enzyme adenylyl cyclase. So how does this relate? This is actually, so what I've just talked about here is really this picture back here that we just looked at, this picture right here. And just so you know, in the upper corner here when it says LE 1110, that means it's figure 11.10 in your textbook. So that's, you can relate to where it's at in your textbook. So this is figure 11.10 in your textbook. So what have we just looked at? Here's your epinephrine. Notice here, signal molecules such as epinephrine. Epinephrine binds to the G protein linked receptor, activates your G protein. That was the next step in that picture, or in that pathway. Then the activated G protein activates the adenylyl cyclase, the enzyme. This enzyme catalyzes what reaction? This enzyme, according to this picture. Katie?
It produces CAMP, right?
Yes. So yes, it produces CAMP, so it converts ATP to cyclic AMP. And so this activated enzyme causes ATP to be converted to cyclic AMP. Then the presence of cyclic AMP activates your protein kinase, which then activates the next protein, so on and so forth, until you get your cellular response. Question?
And this is protein phosphorylation.
Yeah, this part right here. So this is protein kinase A is going to activate the next protein kinase by phosphorylation.
And then everything before that is specific to epinephrine.
Yes, yes, absolutely. So I wanted to point out this picture here and what we've already talked about, because this, what we're looking at now, is exactly putting into words exactly what happened here. So now we get the binding activates the G protein, which activates the adenylyl cyclase, which is our enzyme. What does that enzyme do? It converts ATP to cyclic AMP. What does cyclic AMP do? Takes inactive protein kinase A and converts it to active protein kinase A. What does that do? It's going to activate the next kinase in the step, so inactive versus active kinase, and then eventually at the end you get inactive glycogen phosphorylase that gets converted to active glycogen phosphorylase. Never heard of glycogen phosphorylase, most likely, but what do you know about it? It's an enzyme, right? It ends in ACE, so this is an enzyme, and glycogen phosphorylase, what do you think it does? That's right, all right, and that's exactly what it does. All right, it phosphorylates, is the proper term, phosphorylates glycogen. And so it becomes in the active shape. So now we have an active enzyme that was not active before. And so what do enzymes do? They speed up a reaction. What reaction does this enzyme speed up? The conversion of glycogen into glucose 1-phosphate. Let's look at glycogen. What do we know about glycogen? We learned about this. It's a storage of sugar. And what is it really? It's a it's a polysaccharide. It's a chain of sugar molecules. So this right here is a chain of glucose. So it's glucose hooked to another glucose, hooked to another glucose, and so on. Remember that, and I'll put dots on either side. Remember that when we have too high of glucose levels in our blood, our body will take that excess glucose and store them as glycogen, long chains of sugar. Do you guys remember the places that we store glycogen? There's actually 2 places in our body that we store glycogen in. Katie?
Was it from the liver?
The liver is one, and the second one? Muscle. And so that's where we store the excess glucose molecules. So what happens is there may become a time where your blood glucose levels are low, and so we need more glucose for energy. And so what happens is epinephrine, it goes through your bloodstream to your liver cells and triggers this chain reaction to happen, activating this enzyme so that now the glycogen is broken down into glucose. So it's breaking all of these bonds right here between all the glucose molecules, and this is where we get the glucose 1-phosphate. These are individual monosaccharides, individual glucose, and therefore can be used for energy. And that right there is the response. So the response is the breakdown of glycogen to be used for energy. And all of this, see how this would not happen without our first signal molecule, the hormone epinephrine. All right? And so that's your response. And this is where we'll stop. It's really like Thursday, guys.